So in this video, we're going to go over how to do our standard plate count, and we're going to start by going over how to use our micropipetter. So this is our micropipetter, and in this example, we're using what's called a P1000, and a P1000 is going to pipette between a minimum volume of 100 microliters and a maximum volume of 1,000 microliters. And so what you'll see that there's a dial on the front, and on this dial there are 4 numbers. The top number represents the maximum placeholder. So that would represent, in this case, the 1,000. The next number down would be the hundreds, the tens, and then the ones. So if I look at this volume that this pipette is set at, it is set at 0500, which means that it's set at a volume of 500 microliters. So what I'm going to do is I'm going to practice with my micropipette practice sheet. And on this sheet, it has volumes that I'm going to pipette. So the first one says 150 microliters, 300, and then 600. And so I'm just going to do one just to kind of demonstrate how this works. And so I'm going to put this down flat, and I need to start with my volume set at 150 microliters. So to do that, there's a locking mechanism on my pipette. So I need to unlock it. And now I can move the wheel freely to change the volume. And so I'm going to change it so that it reads 0150. So that will be set at 150 microliters. And once I get it set there, I lock it so that the wheel's not going to move around as I go to pipette. So notice my volume is set at 150 microliters. That's the volume I'm going to pipette. So when I use my micropipetter, if I push on the plunger, that's the first stop. If I push a little bit harder, you'll notice I can go all the way to the bottom. That is the second stop. The only time you should ever push to the second stop is when we call the blowout phase, meaning when we're trying to get the liquid out of the pipette tip. Every other time, I'm only going to push to the first stop. So what I'm going to do is I'm going to take my tips, and these tips should be sterile. So I should only open this tip box as I need to do my pipetting. So notice that's why it's been kept closed. And I'm going to open my tip box, and I'm just firmly going to push my pipette down to get a tip. Now notice another one fell out. We'll put that in a used tip waste. So push firmly to get your pipette tip on your micropipetter. Now I'm going to practice pipetting this green dye onto the sheet. And so what I'm going to do, again, it's set at 150 microliters. So I'm going to push to the first stop, and I'm going to put my tip inside the green dye. And I slowly release my thumb. I don't want to release too quickly or it's going to slosh in my pipette. So I release slowly, and for me, I kind of wipe the tip on the edge of the container just to get rid of any excess liquid that might be on the outside of the tip. Now when I pipette, I'm going to steady my elbow on the desk, and I'm going to use my other hand to steady my pipette. And so what I'm going to do now is I'm going to push to the first stop, and then to the second stop, and that's my blowout phase. Now after I do that, I'm going to take my tip out of my liquid before I release my thumb. If I release my thumb while my liquid is still in the tip, it's going to suck it right back up. So I take my tip out of the liquid, and then I release my thumb. When I'm done, I have a used tip waste beaker, and there's an eject button on my pipette, and I'm just going to eject the tip into the tip waste, and then I'm ready to change my volume to 300. So I would unlock it, change the dial until it read 0300, lock it, and then I would proceed to put a new tip on and pipette the next amount. And so that's how we're going to use our micropipetter. For today, our micropipetter is going to be set at some points at 100 microliters, and at other points it's going to be set to 1,000 microliters. And so this is how we use our micropipetter. So now I'm going to do my standard plate count. And so what I'm going to do first is I need to make my cereal dilutions. And so I have 3 of these tubes that are my water blanks. They have sterile deionized water in them, and they contain 9.9 milliliters of sterile water. So I have 3 of them. And if you refer back to the diagram that shows our dilution series, you'll notice that we have one that we need to label 10 to the negative 2. So I've labeled it 10 to the negative 2. Whoops, that's my 10 to the negative 6. Same idea though. So 10 to the negative 2, 10 to the negative 4, 10 to the negative 6. Now, I also have my stock solution. This is my E. coli that I'm trying to determine my titer. So when I go to use this tube, I need to make sure to vortex the tube to make sure that the E. coli is evenly distributed. Now, in this case, we've always taught you that you can vortex by hand, right? But the alternative is that I can use my vortex machine and I just simply push down and it's gonna mix my sample for me. So I'm gonna put that aside for a minute while I get my tip and get my pipette ready. I need to set my pipette at 100 microliters. So I need to change it. So I unlock my pipette and I'm gonna turn the dial until it reads 0100 and I'm going to lock it. So now I have it set at 100 microliters. So I'm going to take my tip and I'm again only gonna open the tip box when I'm ready to do this. I'm gonna take my tip out. Now, before I go in and draw up some of the liquid, I need to vortex this tube. So what I'm gonna do is I'm going to vortex, just kind of mix it. I'm gonna turn it up a little bit. I need to mix it well. And then I'm going to hold the cap in my pipette hand. So I take my cap between my pinky and my ring finger. I'm gonna flame the opening of the tube, put my pipette in, slowly release my thumb to draw the liquid up. I kind of wipe the edge of the pipette tip on the edge of the tube just to make sure that there's no liquid on the outside. Flame it, go back in. Notice that I go under my pipette every time I do this. I don't want to go over my pipette because if I go over it, there's a risk of contamination. So I do everything under the pipette so that I don't risk things falling on there. Now, I'm going to take my tube that I labeled 10 to the negative 2, and I'm going to take the cap off. I'm going to flame it, and I'm going to add the liquid, pushing to the second stop to get all the liquid out. Flame it, cap, tip eject, get rid of my tip. So I have my tube done for my 10 to the negative 2. Now I need to take 100 microliters of this and transfer to my 10 to the negative 4. So I'm going to get a new pipette tip and I'm going to put some pressure on there. Notice that I moved it within my rack so that I know that I've already done this sample. So I know that I'm going into this one next. So I have my new tip on. I'm going to vortex just to make sure it's mixed well before I take liquid out of here. Because again, we don't want the bacteria settling. So I mix it. I'm going to take the cap off, flame it, go in, release my thumb, draw up some of the liquid, flame it, cap. I'm going to add it to my 10 to the negative 4. Flame it, add it, push to the second stop, flame it, cap. Get rid of my tip. Ooh. Hopefully your cap doesn't fall off like that. Get a new tip. So I'm going to get a new tip. I'm going to now transfer from this tube to the next one. So I'm going to take my 10 to the negative 4 and I'm going to vortex to mix it. I'm going to go under, take the cap off, flame it, draw the liquid up, flame it, cap back on. I'm going to add it to the one that's labeled 10 to the negative 6. So I'm going to take my cap between my pinky and my ring finger, flame it, add it, flame again, recap, get rid of my tip. So after I do that, I'm going to vortex this one to mix it. Now I have my cereal dilution. I am done with my bacteria, so I'm going to move that out of the way. I don't need my bacteria anymore. Everything else that I do is going to come from these tubes here. And in fact, my 10 to the negative 2, I'm not going to use anymore. When I do my cereal dilutions for my plating, I'm only going to use my 10 to the negative 4 and 10 to the negative 6. My 10 to the negative 2 was only to make the 10 to the negative 4 and then make the 10 to the negative 6, but I'm not actually going to use it. Now, what I'm going to do is I have a series of 4 plates. And if I look at this plate, you'll notice I labeled it with my initials, so JW. I put Monday, Wednesday, AM, so that would be like if that was my lab section. I put today's date, 9/4/20, and I labeled this 10 to the negative 4. That's the final dilution on this plate. Now, what you'll notice is when we have our empty plates, normally you wouldn't do this, but for the purpose of this, there is a lid. Notice the lid is much shallower, and then there's the base, which is a lot deeper. So you have to make sure when you make your plates that you have the base side down and the lid up. You want to pour your agar into the base and not into the lid. So I arrange my plates, my 10 to the negative 4, 10 to the negative 5, 10 to the negative 6, 10 to the negative 7. I'm arranging them around the flame because the flame is my sterile field. So when I pour those plates, I want those close to the flame. So that's why I've arranged them there. Additionally, when I pour those plates, I need to make sure that once I pour them, that I don't move them after they've been poured. So wherever I'm going to pour them is where I need to leave those to sit for probably about 20 minutes just to be safe before I take my 4 plates and stack them and then put them agar side up and in the incubator. So this is our setup, and now I'm ready to get my agar deep. So now I'm ready to pour my plates. So what I have is I have a water bath, and the water bath is set to 55 degrees Celsius. And the reason that it's set to 55 is that I need the temperature to be kept above 40 degrees because if we allow the agar to get to 40 degrees, remember that it's going to solidify and it's going to harden. So the water bath is set at 55 to keep the temperature above 40 so that we keep it a liquid and we can add the bacteria to it. So what I'm going to do, I have a paper towel to wipe off the tubes because a lot of times they end up fairly wet after coming out of here. And I have my can as an easy way to just hold my tube. So I'm going to come over here and I'm only going to take out 1. And I'm just going to wipe the excess water off of there so that water doesn't spill into my plate. So I'm just wiping the excess water off the outside. Now I'm going to keep it in my can. I need to work quickly once I take this out. So again, I have my 10 to the negative 4, 10 to the negative 5, 10 to the negative 6, 10 to the negative 7. My pipette is already still set at 100 microliters. So I'm going to do my 10 to the negative 5th plate first. So I just take my tip, add it. I need to start with my 10 to the negative 4, and I need to add 100 microliters to pour that plate. So I'm just going to vortex to mix. I'm going to go under, so cap between pinky and ring finger, flame it, and then draw up 100 microliters. I'm going to flame it, cap back on. Now I need to add it to this tube. So I'm going to flame it, add it, flame, recap, get rid of my tip. So I'm going to vortex just a little bit. You don't want bubbles, so be careful. I'm not going to flame the opening of the tube right before I pour it, or actually I can, but what I'll do is I'm just going to flame it and I'm going to let it sit for a minute but close to the flame. And the reason for that is if the rim of that tube is too hot and I pour my agar that has my bacteria in it, that is going to interact with the heat on the rim and it might kill the bacteria. So I like to let it sit for a minute, and then I just open my plate and I very carefully, very slowly Pour it. Try and get all the liquid out. Notice I only open my plate like a clamshell. Flame it, put the cap back on. I'm done with that one. Now I'm ready to get another agar out and repeat the process. So I'm going to take another one out. And I'm going to wipe off any excess water. I'm just going to go ahead and put that in the same can, it's okay. I'm going to do the other one that's going to require the 100 microliters. So I'm going to do my 10 to the negative 7. So again, I'm going to put on a new tip. Then I'm going to take from my 10 to the negative 6, but I'm only transferring 0.1, which is why that's the 10 to the negative 7 plate. So I'm going to vortex to mix before I go in. Flame it. Put my tip in. Draw my liquid up. Flame it. Cap. Now I'm going to add it to this tube. Add it. Flame it. Cap. Get rid of my tip. Now before I pour it, I'm just going to gently, ever so slightly vortex. It will kind of mix when you go to pour it, so don't be too vigorous with that. You don't want a bunch of air bubbles. I'm going to get ready to pour, so I'm going to flame it. And again, I'm going to let it sit for a minute so that the opening of the tube is not too hot when I go to pour it. So now I'm going to pour it in. And then I'm done. So I've poured 2 of my 4 plates. Now I'm ready to change my pipette to be set to 1,000 microliters. So again, I'm going to unlock this and I'm going to turn the dial until it's set at 1,000 microliters. And I'm going to lock it. Okay, so I'm done with these. Let's see if I can get this to fit. I'm going to move these just kind of out of the way for a minute. So I need to now do my 10 to the negative 4. So I have my pipette set at 1,000 microliters. So I'm going to take my tip. Actually, before I do that, let me get out my agar deep. So I have my tube. I'm going to dry off the extra liquid that's on the outside because again, I don't want that to fall on my plate. Okay, I can put it in the can. Now I'm going to take my pipette and get a tip. And I'm going to take from my 10 to the 4 and add 1 milliliter to that tube. So I'm going to mix it, take my cap off, flame it, push to the first stop, put my tip in there, slowly release my thumb, especially with the higher volume. I'm going to flame it, cap, and I'm going to add it to the agar. Flame it, cap. I'm just going to vortex like this for now, just so I don't get too vigorous. I'm going to flame the opening of this. I'm going to let it cool for a minute, and then I'm ready to pour my 10 to the negative 4. Notice that once I pour these plates, I'm not moving them, and I poured them close to the flame so that they're in the sterile field. So now I'm ready to do my last one, which is my 10 to the negative 6. So I'm going to take that one out, I'm going to wipe the outside again to get rid of all the excess liquid. Put it in my can. Get a new pipette tip. I'm going to take from my 10 to the -6. Okay. I'm going to take the cap between my pinky and my ring finger. I'm going to flame it. Push to the first stop. Release your thumb slowly. Take the tip out. Now I'm ready to add it to my agar. So I'm going to flame it, put the cap back on, get rid of my tip. I'm going to vortex ever so slightly. I'm going to flame the opening of the tube. I'm going to let it cool for a minute close to my flame. And then I'm going to pour. Again, you have to make sure that your lid side is up and that the base is at the bottom. So I pour it, flame it, cap. And then I'm done. And so what I'll do is I'm going to let those polymerize, meaning let them solidify for about 20 minutes. And then after 20 minutes, they will be hardened. And I can take my 4 plates and stack them in a stack, and I'll put a rubber band around them, and then I'll turn them agar side up, because remember we always incubate our plates agar side up. So I'll turn them agar side up, and I'll put them in the incubator at 37 degrees. And so this is how I do my standard plate count.